right? So what I want to do, I want to try and summarize it for you in a simple manner, because if you read your reference book or textbook, whatever you want to call it, it's going to be hard. It's going to be too much information. So what I want today is to tell you exactly what to study so that you can really scan this through this chapter in an easy way. This is going to be, I mean, I could have made it the toughest lecture you could have ever imagined. But I tried to simplify it for you. So let's go through what kind of things you need to know. So if you focus on it, in your TBL, the third TBL, I will be asking a few questions. I'll tell you the kind of format we would expect. In your midterm and your final, the kind of questions you would expect. Okay? So these are the objectives. It's all about carbohydrates. This is my contract with you. So if you get a question, that includes the midterm. If you think that question was not part of the objectives, was not explained, challenge us and we can delete it right away. As simple as this. Even for the final, send the email. Among you guys, whoever is representative, could just collect all these objections, send it to us, challenge us, and we'll take it up. As simple as this. We're not here to torture students. We're really here to help the students. But unless they help themselves, we cannot help them. Okay? So, what's the function of carbohydrate? We already know what's the function of carbohydrate. They're a source of energy. They're part of structural components, protein, glycoprotein, cell-cell signaling. They're basically structural components of cell walls and exoskeleton. Did you have the glycoproteins lecture or not yet? Okay, he'll talk about glycoproteins. And that's basically just an idea about glycoproteins. We'll elaborate more. When I say, could you keep quiet please? When I say cell-to-cell signaling, you can see that if you have here a receptor, transmembrane receptor, we're going to discuss these in mole 125, hopefully once you pass 114. You'll see that there's a portion of it which is carbohydrate. So we have an oligosaccharide chain attached to the protein, so they are part of the cell signaling with the viruses, with bacteria, with lymphocytes, cell-to-cell interaction, toxins, glycolipids, also they're there. If I tell you that 50% of the proteins are glycosylated, so they are part of the glycoprotein, 50%. Do you know the percentage of genes involved in carbohydrate metabolism of your human genome? Carbohydrate, 1%. 1% of your genes are involved in carbohydrate metabolism. That tells you that this is really a significant component of our lives. So the whole idea here is just to show you that they're part of glycoproteins, glycolipids, and they are involved in different functions. So let's get into some bioorganic chemistry. We know the carbonyl group, right? So we do classify the carbon, the monosaccharides. We know what's the monosaccharides versus polysaccharides, right? Yeah. Polysaccharides can be hydrolyzed to monosaccharides. Yeah. To break it down, right? This is how we store our energy in terms of starch, glycogen, and so on. What's the oligosaccharide?
10.
2 to 10. Would you classify disaccharide as oligosaccharide?
Yes.
Oh, disaccharides too. We're talking about 2 to 10. Yes, they're part of oligosaccharides. Right? So, we have the carbonyl group. So, we do classify the monosaccharides based on the location of the carbonyl group, as we will see, and also the number of carbons, trios, pentos, hexos, and so on. And these basically could form, the carbonyl group could be part of an aldehyde or ketone. What's the difference? 1R group, 2R group. That's organic chemistry, which you all have, right? So, we have two metabolites here. Glyceraldehyde and dihydroxyacetone. Does it ring a bell?
Yes.
They're part of what? Intermediate for what? Glycolysis. I know you have lecture of glycolysis next week. And they will tell you how glucose is broken down to glyceraldehyde and dihydroxyacetone. They're part of the glycolysis you're going to study next week. So are you telling us that glyceraldehyde, dihydroxyacetone are sugars? Yes, they're trios. aldotriose, they are ketotriose. Why? Look at the structure. Aldehyde group, keto group. Right? Can you tell me which group is the chiral group? You know chirality?
Yes.
Is it, okay, we have C1, C2, C3. Tell me which one. C2 is the chiral group. Do we have a chiral group?
No.
We don't. Right? So, let's look into chirality and how it really makes a huge difference in our metabolism. We have glucose, galactose, which are aldoses. We have ketoses, including the fructose. Where do we get fructose? Do you want to define fructose? Fruits. Is it healthy to drink fruits?
Yes.
What do you think? Drink.
No, it's better to eat.
Can your body metabolize fructose
Yes.
or galactose?
No.
Can your body cells metabolize it?
No. They change it to glucose.
We don't metabolize it for energy. Why? So why do we drink it? We absorb it, and what happens to it?
Change it.
It gets converted to glucose by the liver. So when you drink too much fructose, galactose, that also could be bad for the liver because the liver now has to function to convert it into glucose. We could utilize it for structures, for intermediates, but not for energy production. Are we clear? So everything, that's why we only study glycolysis, glucose. We study gluconeogenesis, glucose. What about the others? What happens to them? We break it down to monosaccharides, we convert it into glucose, and now it gets into our metabolism. Right? So that's why we measure carbohydrates, why we measure glucose in our circulation, and so on. But what I want to show you here, chirality. So, we have an aldose, and we have a ketose. What's the numbering here? What's the number for this C?
C1.
Oxidized carbon. C1, right? 2, 3, 4, 5, 6, and so on. What about this one?
C1.
This is, no, this one, I'm sorry, what is this one? This is a ketose.
Yeah.
C1, 2, 3, 4, 5, 6, right?
Yeah.
Now, which one is the chiral carbon molecule that determines whether it's a D or an L?
The fourth.
The bottom chiral center carbon. This one, the C5. Because we're going to discuss D and L glucose. Now, this is the bottom chiral carbon. Is this chiral we said?
No.
No, this is the bottom chiral molecule from the oxidized carbon.
I don't understand the chiral.
Okay. What's chiral? This is glucose, this is galactose. How do you describe them? Are they mirror images
No, they're
or non-mirror images?
mirror images.
What do you think? Let's look at it. They're not mirror images. Right? Look at the most, or the bottom, chiral, central chiral carbon compared to the oxidized. This is the bottom. What happens here? You have, if you look at, this is what we call D, glucose, because it's pointing to the right. Now, what I just want to show you, now we're going to talk about chirality more. If you look into this carbon, what do you think? This is glucose. Is this different or similar?
different.
It's different. That makes this as glucose. This is galactose. This is C4. If you go for mannose, let me just show you an example. Yeah. This is the glucose. 1, 2, 3, 4, 5, 6. Galactose, we said C4 is different. What about the mannose? C2. Look at C2 compared to the C2 of glucose. Now I have 3 monosaccharides. All what it is, they're